I would like to talk a little bit about our experience implementing single step. Since July last year, it's been running every week, and we've been running a weekly evaluation at Angus since about 2010, and that essentially went together with genomics. Once we started incorporating genomics in the EPDs, they went to a weekly evaluation. And a little bit about the company. So Angus Genetics Inc. is basically a subsidiary of American Angus Association. So it's a for-profit company. And I put this slide up because people say, well, what's the future of breed associations? You know, what role do they play? And this is our team, and there's some faces there you won't recognize, but a scientist we've recently hired will join us here a couple Mondays from now. Duke Liu was working for us at Ag Research as well. We went to North Carolina, and now we've attracted him. So I think for a breed association, you know, we have to be at the front end of science. We need to make the best tools available to breeders. It's not just about just registering cattle anymore. And I've got these are you know, Dan Moser's was a professor for a while at Kansas State University. He's basically my boss and the president. We've got Kelly Ritalik. She's on the phone 70% of the time, really, with members about questions about EPDs. Gives a lot of talks out there in the industry. This whole bottom row here are basically all in the information systems group. So these are people, you know, they're writing code. They're behind the computers all day. There's a lot of data that needs to be taken account of, and obviously you've got your own people here that do those things. These are 8 faces that are basically on the phone all day dealing with breeders about their DNA tests. So, you know, we've got about 3,000 samples a week come in from members. There's 25,000 members. That's a lot of phone calls and a lot of time, and these people look after all that. So we do genetic evaluations not just for Angus, but for some other breeds as well, but Angus is the biggest. In terms of what we deal with. Basically there's more registrations in Angus than all the other major breeds combined. So that's to give you some idea. We also, just on the genomic side, we work with partners with 2 companies, basically Neogen and Zoetis, and here in New Zealand, for example, the I50K and soon to be, you know, well, I50K, and then we've got a GeneMax Advantage, which is kind of a commercial prediction for heifers. So a big part of our business now is the genomic testing. So all the samples come to us in St. Joseph, and then we send them out to the various labs and incorporate the results back. So if Time Magazine puts out a year in review, and I thought if I was the editor, I'd change it a little bit. So I changed it a little bit here. You know, we've got, Angus and single step, I think, should have been, and this is our SAR evaluation report, you know, that should have been maybe front page, because for Angus breeders anyways, a little self-centered, it was a big year. 2017 and July 7th were major changes to the EPDs. And, bulls re-ranked, and they re-ranked in the indexes, and there was a major change, and that's what I would talk about that transition. So I think from BLG's perspective, mornings like today are really important. It's all about communication. And we did a lot of communication ahead of time. We were running it in parallel week to week from November through to July. So we could identify, you know, how fast it runs, how stable it is, how much animals change week to week. And we did a lot of work we knew what the changes were going to be. But a bit of a different approach, we didn't actually show those breeding values to producers. So no producer ever saw how the bulls were going to re-rank until we rolled it out. Even the board of Angus didn't see those numbers. So there was a lot of anticipation, and you try and communicate to people, well, it's going to change. Well, how much change? Well, you show them what a 0.9 correlation means or a 0.95 correlation. And I realize now that no matter how much you talk about a 0.95 correlation, they really don't fully grasp how much animals can re-rank with a 0.95 correlation. So then we rolled out the numbers, and although we did a lot of communication ahead of time, and leading up to that stage, I'd say we did more. I was never involved with so much kind of, you know, fact sheets and articles and magazines and things on television and things like that. But still, I had breeders call me up a couple weeks later and say, you know, how was I totally blindsided by this? How could I, you know, never knew this was coming? And I'm like, well, I guess you don't read the magazine, you don't watch the internet, you don't watch television, or any of these other things. And I'm probably preaching to the choir here, because everyone here is probably not going to be in that camp that they weren't expecting any changes coming. But then we kind of dropped into a mode of more communication, and this little box here in the bottom left-hand corner was probably, there was a lot of that, a lot of telephone. Breeders like to call up and talk to somebody, but there's also a lot of things on the internet, fact sheets and things like that. So once kind of the people realized what the change was, and they realized this is it, and this is how the new numbers are, and you communicate that they're better, then they just want to understand, right? They want to understand better, how does the system come up with these breeding values? How can I really trust it? How do I know they're different? You told me that the other ones were good, and now these ones are a bit different, so how do I know they're good? And so you have to work through all that. So there's a lot of communication, but I can say now that was about a month or two of a lot of communication, and then it's just been kind of full steam ahead since then. So for us, it wasn't just rolling over to single step, the statistical method. We actually implemented a lot of things at the same time. And these are maybe some things that have been in development for a while. And the idea was, if we're going to make one change, we'll actually make them all at once. And it's kind of like taking off a Band-Aid. You want to just rip it off, get all the screaming over with at the same time. So, we went to single step, but we also changed our model behind our carcass traits. So it's a better model. It's more comprehensive. We account for more things, which was really important, and we got that implemented. The other thing is, we were in a situation, and this is one of the beautiful things about single step, is this whole idea of doing a calibration kind of goes away, or at least every week now there's new calibrations. So instead of kind of jumping in step changes, you go through this calibration, everyone kind of re-ranks, and then you go through another calibration 6 months or a year from now, or whatever the time frame is. In our case, our calibration was over a year old. And so we had added over 100,000 animals genotyped to the database since the previous calibration. So when we went to single step on 7th of July, we dumped all that data, got dumped in. So that's another source of change, really. We'd also gone through and updated all the heritabilities and genetic correlations, updated the dollar values for the economic indexes. So all these things kind of happened at once. And the breeders would say, when you guys went to single step, my bull changed or whatever. But a lot of times it wasn't due to single step. Some of it was due to, we changed the carcass model, we changed the heritability. These things are nothing really to do with single step. It's just all those changes happened at the same time. So I just, to get across the point, they were relatively big changes. Single step, and we heard this morning we're going to have a whole session on single step. It's really become it's almost like a brand, right? Breeders talk about it like it's a brand. It rolls off the tongue pretty easily, and I think it's not necessarily. I think maybe as the industry, maybe we can get in front of some of these things and give things a name. So I think it's really great. And New Zealand does a really good job with that. N-prove is something I just heard about today. But you're really good at giving things a name, and then people know what you're talking about. But I think the cat's out of the bag as far as single step goes. It's kind of a household name now, or rolls off the tongue with breeders. So we talk about the software and things, but I want to emphasize, and a lot of the posters and things that you see downstairs here and the efforts with beef and lamb genetics, it's about data as well. We can implement the best genetic evaluation software, the best kind of mathematical tools, but it all depends on the data behind it, right? If you don't have good data and enough of it, you're not going to have accurate predictions, and the best statistical methodology in the world is not going to help you. So you can't get away from collecting data. In our case, we hit a major milestone here just a couple weeks ago, and that is we had kind of our 500,000th genotype come into the system. And today I was thinking about it, because I thought I was kind of anticipating the day that, I was kind of see it come, hey, we're going to hit 500,000 genotypes this year. That'll be kind of neat. And so I think that's a big thing. But I was wondering a couple years from now or 3 years from now, I'll look back and say, geez, I thought 500,000 genotypes was a big thing. But It's not a big thing. You know, if this keeps ramping up like it is, soon that won't be a big deal anymore. I just look back in time, I said, well, how long has it taken our genotype database to double? And although genotypes go back to 2010 or 2009 and that type of a time frame in our database, we've doubled the database in the last 96 weeks. So when I was first approached about would I be interested to come and work at American Angus just about a couple years ago, our database was half the size. And so it's been increasing. And I wanted to show this slide, not so much to talk about 500,000 genotypes, but to actually talk about, well, what's been the reaction from the breeders? So we rocked their world, really, with, we changed the ranking of bulls. Some bulls went up, some bulls went down. There's winners and losers. What was the reaction? Did they kind of throw up their hands and say, all this genomic stuff is a waste of money, I'm not going to genotype anymore? But that hasn't been the reaction, right? If anything, the genotyping rate has gone up. There's been some other things. At the same time, we've rolled out the price of our 50K genotypes have come down considerably, and so that's helped, but we're probably up 20% on genotyping volume. So after the dust kind of settled on, some people angry with their bull that might have changed or something like that, why else did people they're trying to understand what the differences are, and one of the biggest questions we had, or people calling in, was, how can these two flush mates, and I say flush mates, embryo transfer, I've got two embryos, they're born and they don't really have any phenotypes themselves that we use in the evaluation, but they genotype those calves and we give them a different EPD, or in New Zealand terms, EBV. How can they be this different? So with single step, basically, we're putting a bit more emphasis on the genomics, and we're actually spreading out those embryos more than we had been in the past. So they hadn't seen this kind of spread before in embryos. And so they look at this and they say, well, are you sure this thing's working right? Because I've never seen this kind of difference in embryos before. And so then we did a bit of analysis in that area. So as I was working on the phone one day with a breeder about, why there was this difference in his EPDs and everything, and explaining to him about what we're going to talk about with Mendelian sampling, and it's a chance when you just happen to inherit more from this ancestor than other ancestor, et cetera, et cetera. And if I quoted him directly, it's something like this. He goes, so you're telling me that this breeding is more of a crapshoot than I ever thought it was? There's more luck involved than he ever thought was possible. And well, actually, yeah, there's a lot of luck. Every time, whether you call it luck, it's random variation. Every time that sire produces another calf, he throws out another random sampling of his genetics, and sometimes it's on average better dose or a worse dose, and that just happens over and over again. So there's just a lot of variation, and that's what I want to get across today. So what does single step really do? Single step's really all about this pedigree, right? And so we'll talk about the pedigree, and I've got a picture of a bull there. That bull, Traveler 23-4, is the most influential bull in the Angus pedigree. So we estimate that 9% of all cattle born today 9% of their genome comes from 23-4, on average every calf. So it's hard to find someone who is not in his pedigree. And I'll show you a pedigree in a minute, and he's in there a number of times. So there's a lot of connections in the pedigree because there's been a lot of AI use, right? So because of a lot of artificial insemination, there's a lot of connection in the pedigree, and so you send in pedigrees as breeders. You send in a pedigree that looks like this. We got a calf here, X, he's from this sire and this dam, and then the sire's pedigree was this paternal grandsire and this paternal grand dam. We've got this little pedigree. What's happened behind the scene is your scientists here, when they do your genetic evaluations, they convert that file into something that looks like this, and we call this a matrix. This is the relationship matrix amongst those animals. How related is this calf X here to his sire? Well, on average, he inherits half his genetics from his sire and half from his dam. And how much does he inherit from his paternal grandsire? Well, another half, because every generation it halves. And so he inherits a quarter from each of his grandparents. And that's all we can do with the pedigree. That's just how it happens on average. But we know that average is good on average, but it doesn't apply to the individual. So, an individual could actually have inherited between, this was, there I was working again, this paternal grandsire and this paternal granddam, he might have inherited a bit more from this side and a bit less from this one. And with the genomics, with the 50,000 SNPs, we can track that. And so we can actually change those numbers. And if going back to graduate school and working with genetic evaluation, this matrix is kind of the glue that holds the whole thing together. How can we compare animals in one herd to animals in another herd? It's because they're connected with this relationship matrix. So we've got this relationship matrix that crosses all the animals. So if we do an evaluation with 10 million animals, we've got this big 10 million by 10 million relationship matrix. But with the genomics, we can actually, instead of putting in the 0.25s, we can plunk in what the relationship really is based on the DNA markers, and it's more correct, it's more accurate, and that's what basically, as we kind of twist this A matrix to reflect what the relationships really are, we change the EBVs that come out the other end, and that's what single step's doing. So as I work through with breeders, how can two full sibs be different? We start talking about this whole idea of Mendelian sampling, and this goes back to basic biology in school that you would have seen, but you just to think about the principles. We've got two chromosomes here. If this is a paternal, this is a potential sire. He's got two chromosomes. One he inherited from his mother that was in the egg, and from his paternal granddam, and the other one he inherited from his sire through the semen, and I've got them here in blue and pink, which kind of universal colors, I think, to keep them separate. But this sire now, when he's going to put out a dose of semen to create a lamb, say, what's he going to send? Is he going to send what he got from his sire or send what he got from his dam? And of course, he doesn't send either of those. He actually sends a mixture of both. And so in this case, we get what we call a recombination event right here in the middle. And he's going to send on a sperm cell that actually carries, the top half of that chromosome they inherited from his mother, and the bottom half they'd inherited from his sire. And he might pass that on to this calf here. And the green chromosome over here is what this calf got from its dam. And I didn't show that, but just put the green chromosome in to give you an idea. So that's what could have happened. But then he produces another calf. And it could look something like this. We don't have a break in the middle, we have a break at the top. And we pass on this piece of chromosome. And now this calf, if you can see, on that chromosome he inherited a lot more DNA from his paternal granddam than he did his paternal grandsire. And we're always looking back at the grandparents and great-grandparents, because you always get half from your mom and dad. But your mom and dad is how much of each of the grandparents do you pass on. That's where the variability comes in. And I think as breeders, this goes back to what breeders have talked about for years. I remember looking at a particular bull with a breeder, and he had great EPDs, but the breeder looks back at the pedigree and he sees some sires back there he doesn't like. Oh, he's got that bull back there. He was good or bad for this and that. He's worried about those things. In reality, those animals way back in the pedigree, you might have inherited more or less from them just by chance. Just by chance. And it's in this with genomics that we're actually tracking those things. So this is just another possibility. Here's three calves, and they've inherited different amounts of DNA from those ancestors. And the genomics is going to track that. And it's going to change that relationship matrix to reflect it, which is going to change the EPDs or EBVs that come out the other end. So this is not new. This is old stuff. It's been around for a long time with what we call Mendelian sampling. So I'd like to talk a bit about variation, and breeders talk about variation different ways, but usually they think about, they want to make things uniform, right? The industry wants things uniform, but variation is actually the tool that you can use as breeders to make change, right? And there's always this, the good news is most of the variation that we see is not genetic. It's to do with the environment. So if you really want to make uniform chickens, put them all in the same facility, feed them all the same, and you have very standard management, and you can decrease a lot of variability, keep your animals healthy, decreases variability. So we can do a lot with environment. But just to talk through some principles about variation, here we have birth weight in pounds, so roughly divide by 2 to get kilograms. That'd be kind of 25. These are both calves born in our Angus database, run 54 to just over 100 pounds, and they average 78. And that's the real bell-shaped curve with our data that comes in. Now, we know that most of that variation is due to the environment, because some are born in cold environments, hot environments, and different feeding, different times of the year. So there's a lot of environmental variation, but over 40% of that variation is due to genetics. And so we look at how much variation then do we have in the actual EPDs, and they run from minus 7 to plus 9 is the EPDs that go out, and that's the variation. But what's the source of that variation that we see? And if we go to kind of, again, back to standard economic theory, there's 3 sources of variation and how much variation there is in those calves. The one is just how much variation is there in the sires that created the calves. So if they were all from the same bull, which they're not obviously, because that's all the calves born in the last year, that'd be 25% of the variation. 25% of the variation is due to the variation in the mother's genetics, but half of it's due to just that Mendelian sampling, the random combination of sires and dams. So half of the variation that we're dealing with on a genetic basis is that part that we're getting at now with genomics that we couldn't really get at before. So back to the breeders' questions when they call up and they say, well, I've got, how can these 2 ET calves be this different? And they said, well, let's pull up all the calves that were born to ET. At the time, there's 646 of them that came from what we call commercial recipients. So they're not registered Angus dams, and we register the calves, but we don't use their records because we can't really properly account for the dam and the evaluation. So we've got, they were all genotyped, and we put out EPDs on those calves. And what I've done is I've centered this all on a +2, so I've taken the average of the parents' EPDs, and so I could line them all up on the same scale. If the parents were both +2 for birth weight, which is just about breed average, this is what the distribution would look like. And I was really happy with the graph to start with because it's looks pretty normal, right? It's got a distribution that looks reasonable. And these are actual EPDs that went out the door to breeders. So with the same mating, we sent out essentially the same mating. We sent out some animals that were +5 for birth weight and some animals that were -1. And from a breeder's perspective, if we have calls from breeders, I'd say 65% of the calls are something to do with calving ease and birth weight. It's kind of a no-go point to sell a bull in the U.S. People are kind of laser focused on calving ease, and they just won't tolerate. Once you get to a certain level, they just won't look at the bull. And that generally starts around 3 to, depends who you talk to, but once you get over 3-4 pounds of birth weight EPD, it's hard to sell the bull. So that, when we could have given you a -1 or a +5, you're basically saying he's super calving ease bull, or he's, you shouldn't have kept him as a bull. And so that's the difference. It's a huge range and has a big economic incentive or a big economic implication. So I'd like to, there's a lot of movements going around, so I thought I'd start my own movement for breeders out there that, I heart variation. That's really that. We go back to the slide, and, the breeder that got the bull that was +4, he's not happy, but the point you have to talk to him, well, isn't it better that you know now? Isn't it better that you sold him than you would have sold him to someone that would have bred him to a lot of heifers and he would have had a wreck? Isn't it better that you actually know? And so this is the tool, right? The idea is now at birth, we can identify this variation in full sibs, and you can select the best ones and get rid of the rest and not waste and have that heartache of problems down the track. So what's happening, we just had a Beef Improvement Federation meeting, and one of the presentations was from a company there that's doing a lot of embryo genotyping. So, he's creating IVF embryos, and he's taking a biopsy. He's genotyping that biopsy, and then we run genomic predictions on those biopsied embryos. And he's just got to the point now, and this was last week, they wanted because they're putting together a sale catalog for a pregnancy. So they've got one of these embryos. They've got a bunch of the embryos, but they've got, here's a cow, and she's carrying this embryo, and the embryo's kind of off the charts for some of the traits. You know, they're the highest growth EPDs that we've ever kind of put out. And that's kind of where the technology is moving, right? This is some of the things that you can do with it, and it's actually happening now. So the question from breeders is, you know, a picture didn't show up there, but that's okay. The question is, if I mate, you're telling me if I mate a +2 cow to a +2 bull, that you're going to give out a +5 EPD. Is that real, or have you guys made a mistake? And the answer is, based on Mendelian sampling, it is real. And to have a look at that, I said, okay, let's do a little simulation based on theory, so I know what the genetic variation is in Angus. And if I applied standard theory and I simulated 1,000 progeny, if I had a +2 mating, and I simulated 1,000 progeny, what variation should I see in the EPD? And this is kind of my variation that in theory says I should have. It's running from a -3 up to a +8. So I actually simulated more variation than we saw in the EPDs we put out. And the reason that is because the EPDs we put out are not 100% accurate. Right? Our genomics isn't that good yet that we actually predict all the genetic variability. We're only predicting some of it. They're still, you know, they're accurate and useful, but they're not 100% accurate. It's not the same as a highly proven sire that's got 100 progeny or something is 99% accurate. So we've got more variation in their true EPD than what we score. So in reality, for the breeders, you're mating +2 cows to +2 bulls. You are creating some +8s. Right, they are going out there, and if we progeny tested every calf born, we'd identify them. But at the moment, that'll wait to be seen. But so it is real, and the variation that we're putting on the calves seems to be real, and it follows what the theory says it should be. It's just bigger than what the breeders had seen before. But that's essentially the root, I think, of progress. So Mendel says I'm going the wrong direction. Sorry. Mendel says we're doing a good job, and it's what the theory says it should be. So this is an example. Again, 2 full sibs. They're both born from embryos, and they've got a different EPD. And the question is, does it kind of match what we might think when I look at the pedigree? And so I went through, and this is the top side of this pedigree, and if I know there's some Angus breeders in the room, so they'll recognize some of these names. There's lots from this bull, so that you can't tell what breeder it's from or anything like that. But so I went through and I took this kind of 6th generation pedigree, and I said, how is the animal related to each of its how are the 2 embryos related to each of its ancestors? Based on the pedigree, it'd be the same. But the genomics tells me it's different. And does the difference in the relationship actually match the difference in the EPDs? That is, if the calf that had the higher birth weight EPD, is he more related to the ancestors that have got higher birth weight EPDs? And so you kind of do these things holding your breath so that hopefully that all comes out as you hope it would. So what do we think about on average? So here's this calf. His sire goes back to this bull, Shearbrook Shoshone, and I had somebody correct my pronunciation because I wanted to call it Shearbrook Shoshone, but apparently it's not pronounced that way. So Shearbrook Shoshone, you know, he passes half his genetics on to his son there, another half to New Trend, another half to New Day, et cetera, down the pedigree, and his sire, Reserve, passes on a half. So we are 1/64th of that Shearbrook Shoshone should be in the calf. That's the relationship we'd expect. It's a bit different because some of these sires show up in the pedigree multiple times, so we have to account for all that. So we did this for all these ancestors, and you see them listed here, and I've got them ranked by their birth weight EPD. So I've got, here's the bull in the pedigree with the biggest birth weight EPD, was a plus 5.5. So I already told you, if you were to try and sell that bull today to most people, they'd say, they're not interested. But he's back there in the pedigree, and he's plus 5.5, and he's in the pedigree once 4 generations back. Here's a bull. Where's Power Play, for example? Power Play is just below average for birth weight EPD, and he's in the pedigree 5 times. So he's actually in that pedigree 5 times. So we add all that up and we think, okay, how much of grid maker do we expect in the full sibs? Well, we expect 0.0625, but what we find is one full sib actually has like 0.03 more than the other, which is, these are small fractions, but they all add up, right? We added up across all those ancestors. That's a 50%. So we've basically got 50% more of one ancestor than the other in that calf. It's a small amount, but it adds up quite a bit. This is kind of an extreme example. This Rito 2100, there's twice as much of him in the one full sib than the other, as compared to what we'd expect. So they're small numbers, but still, these things add up. And these are absolute differences. Just to give you an idea. So what I did was I said, okay, let's take all these ancestors. I ranked them by their birth weight EPDs, and let's look at the relationship from the heaviest. These are the bulls that are above average for birth weight. These are the ancestors above average for birth weight, and we add them all up. And what we find is the sib that was the bigger birth weight EPD was 15% more related to those high birth weight ancestors. The bull that was, and he was 13% on the other hand. Obviously if he's more related to these, he must give it up somewhere else. He's actually less related to these low birth weight ancestors on average. So it doesn't hold up perfectly. If you look at up here, there's some positives and negatives. So it's not, but it's just how it all adds up is basically what contributes to his EPD. So I had a breeder after giving this talk, and a breeder come up, and I think this is essentially what breeders have been wanting to get at for a long time, right? This is what breeding's all about. I'm going to use the sire. He's got some ancestors. Some of them I like, some I don't like. Let's make some progeny. I want to get the progeny that carry all the bits I do like, but aren't carrying the bits I don't like. And single steps is actually giving you a tool to do that now. So one of the phrases that we're kind of using, explaining things as the rollout to members with single step, was a quote, and I looked it up. It's by nobody in particular, kind of an anonymous quote, but different's not always better, but better's always different. And so we rolled out single step. We did a lot of testing, and just like your team has been doing, when I showed up at Angus, essentially they basically already had it pretty much up and running. So we were kind of 6 months of testing, and a big part of the testing was, okay, we got these numbers are different, but are they better? And so we did a lot of accuracy predictions. You got that red card up yet? I don't know. Are you waving the red card yet, Becky? 2 minutes, okay. So we did a bunch of work to say, well, it's different, but is it better? And what we found was the predictions with single step better predicted future performance of the calves than what we were doing previously. So that was good news. We did some work in other populations to say, okay, the numbers we're putting out compared to what we were doing, how does it predict these independent populations somewhere else where the data is not in our population? And single step basically did better. In our case, there was, we updated the models, we did a lot of other things that made it better as well. So, but if you think about what's the biggest change with single step, it's really the beauty of it for us week to week is that it's always current. All the data's in there. So every week as new genotypes come in, all those genotypes, all the pedigree, everything, the number that goes out the door incorporates all the data real time. So we don't have this, well, we got this data in, when we do a calibration in a few months, it'll go in there, and then, then it'll influence your EPD. So it's always current. Maybe another point I'd like to make is, what genotypes can we use to influence the pedigree? They're these 50K genotypes that influence that pedigree relationship matrix. So when I showed 500,000 genotypes, those are 500,000 50K genotypes or imputed to 50K from a lower density test. We have about 1 million samples. We keep all our samples. We have 1 million samples, and all those samples came in for something. So there was a lot of parentage genotyping and defect genotyping and things in the past, but now we're basically moving everything to a SNP chip where it's genomic selection. You obviously, and all your parentage and defects and things are just part of that. And so that's kind of where we've gone to. So to actually do single step, you need to have 50K genotypes or something you can impute to 50K genotypes. That's what it's going to use. So single step, so it's really all about relationships, but I think breeders have been all about relationships for a long time. And single step is just using the markers to develop a better relationship to go into the evaluation. And with that, I'll turn it over for questions.
Yeah, thanks for that, Steve. I thought that was an excellent explanation, just showing the importance of the pedigree further back. We all focus on the parents, whereas really the grandparents and the great-grandparents have got just as much to play. So we've got about 10 minutes for questions. If you've got a question, please put your hand up and wait for a microphone to come to you. So, got one over there.
Thank you, Steve. Just one thing, sometimes you get a ram or a bull that sits at the top of the list genetically. He's a very strong producer and he stays there. Is it possible that he's got more genetic grunt in his genetic makeup, that when it passes, more of his gets into the offspring than the other side, the female? Are they just better at that as well, not just the genetic, but also the transfer of that genetic information?
Yeah, there's a little bit of what you're getting at with your question is something that we on the genetic space, and we've actually got working with another researcher on a project around this. It's called transmission ratio disequilibrium, right? Where we have these basic rules where you're always half of your mom and half of your dad. But are there cases, and they've shown, so there's been a lot of research in mice and pigs and other things to show that this transmission ratio, that there can be a disequilibrium in inheritance. And so they've shown these things in other species. We're not, it's a new area of research to see how real is that in cattle, and how could it be influencing our EPDs, and that we don't necessarily, we don't know yet. But how big it is and how real it is, we don't know. It's potentially an area.
So what's your hunch though? Do you reckon it's happening? Well,
so one of the projects that we're working on at the moment is really we're using it for predictions related to fertility, and in dairy cattle it's well known, where we've got regions of the genome that are fairly frequent, but they never exist kind of as two copies in any individual. So we genotype 500,000 animals, and we've got this region of the genome that we know is floating around with a reasonable frequency, but we never see it in the homozygous state. And the question is, why not? And the reason is because when we get that homozygous state in an embryo, the embryo basically doesn't live. And so that's a biological reason. That embryo then basically, the cow absorbs that dead embryo, she recycles, and then you breed her not 21 days later, but 30 days later or something like that. So that's actually a biological mechanism that can control some of this disequilibrium. So that's stuff that we are dealing with right now. We haven't we've got to validate them. We've identified the regions, but we're working to basically validate them before we roll them out. Then it can be a tool for breeders to use for breeding for better fertility, actually.
Talking to it.
Okay, there you go.
Steve, I'm just interested in how you factor in the environment. And we hear a lot about epigenetics and the fact that the environment and extreme environments particularly can trigger or activate different genes. And this is the area epigenetics where these genes are sitting there unactivated normally, but under stress they get activated and then they have the ability to pass that on to the next generation. So do you, in your genomics, recognise those genes sitting there not activated, or how does that work?
Yeah. Right now, so there's no there's no accounting for those epigenetic factors in the evaluations that we do at the moment. Environment is essentially all accounted for through this what we call contemporary groups, right? So you've got this group of calves that are all raised on the same farm in the same environment, weighed on the same day, and we've got data edits just like you do to account for all those things. The whole field of epigenetics is, had a producer in my office the other day talking about that. There's a lot of articles and press about it. It's fascinating science, and a lot of it happening here in New Zealand, around that area. At the moment, it's just not influencing breeding values for us, but it's not influencing breeding values right now for anybody. Like, the pig, the poultry, the dairy guys, that unless there's been a change here recently, nobody's really accounting for it in the genetic evaluation. I think it's this current area of research. In the future, it could do. One of the tools and that we need to basically use it in evaluation, have it start to influence our evaluations, is basically a chip where we can genotype those regions to get at those epigenetic factors. And that's possible, but at the moment that's not something that we've got at our fingertips. So, and there's some researchers and initiatives here in New Zealand and elsewhere that would like to put one of those chips together, because we got to get a lot of that epigenetic data to match it with the real data in the field before we can. So, from a producer's point of view, to say you're going to account for epigenetics, it won't be next year, it won't be the year after that. I think it's possible in the future, but it won't be in the next couple of years. Is my prediction on that. Yeah. So right now our breeding values are as accurate as they can be, accounting for the average environment. If we could actually use epigenetics to account for the fact that within that group of calves that were all raised the same and born in the same place, if this one over here was born on a rainy day in a mud puddle, and this one over here was born in beautiful sunshine. We can't account for that right now. But in the future, with epigenetic chip and a lot of data, that's a potential development. But right now, we just count for that average environment. If that wasn't good, if what we were doing wasn't good, our heritabilities wouldn't be very good. So just accounting for the environment like we do, we have a heritability that we can actually make some progress with. Not to say it can't be better, if we could account for those factors, but.
Just one more question. Yeah. Two. Yeah. Over here first, please. Here on the left first, please.
When I heard the 500,000 genotypes, my heart sank, because I thought, how the hell are we going to get anywhere near that with sheep? But it's the proportion of the population, I guess, that are genotyped. So what proportion is that 500,000 of the animals that have phenotypes recorded as well?
Yeah, very good question. So it depends on the trait. Right, it depends on the trait, because typically these are there's obviously they're seed stock animals, they're registered animals, and so when the animals, birth weight, weaning weight, yearling weight, ultrasound scan data, that's depending on the animals, but most bulls, I'd say at the moment about a third of calves coming in are genotyped. And it's a bit more in bulls than heifers, but not a lot more. So we get a lot of genotyping done in heifers, which is really good, because then we get heifer pregnancy records and things like that. So really the database kind of builds itself. As guys are genotyping, those same animals come through the system and they get phenotypes, and that's really the beauty of week on week. It's just as the data comes in. So it's a bit different than developing a reference population, where we genotype and phenotype and then people just genotype to get a prediction. the people genotyping for the prediction are also contributing the data in our system.
In our sheep industry, where we've got a lot of smaller breeds that we can't even genotype for, so the best we can do is just do lots and lots of phenotypes. Lots and lots of actual measurements, because...
Yeah, well, kind of a message we try and get out to producers, just with the way single step actually works, is again, it's related to relationships. So in a way now, a producer can kind of almost build his own reference. You know, that you're collecting your own data, you're doing your own genotyping, and so when you bring a new animal in the system, it's how is that animal related to those other ancestors that you phenotyped and genotyped. So, you know, we've rolled out, I think single step has actually started to bring genomics into some breeds that didn't have as much genotypes than what they did have. So I don't think, you know, the 500,000, but we're starting on a project right now for a health trait, and our target to start with is 4,000 animals phenotyped and genotyped to get started with a prediction. So.
Keep me just one quick question.
Yep. Peter Amer from Abacus Bio. Great talk, Steve. Could you just quickly tell us about those third of the people that are doing the genotyping? What's driving them? Are they big corporates? Are they small mom and pop breeders? You know, what's kind of the driving force that's motivating the uptake of this technology?
Yeah, well, it's kind of right across the board, really. You know, one of the challenges just around phenotyping. You know, right now you saw ultrasound scanning. There's like 2 million scans, but at the moment, our prediction with genomics, you know, the extra accuracy for an ultrasound scan isn't that much. You don't add that. It's a lot of work, but breeders are still keeping up scanning. You know, what we're seeing is maybe scanning's falling off. I realize I'm talking about phenotypes now, but they're kind of falling off in some of the smaller herds, but it's because they're just doing genotyping. So in a way, and I don't know that's the bigger breeders are keeping up the phenotyping. So it's, as far as genotyping, it's actually maybe been as much of a boon for the smaller breeder as it has been for the biggest breeders. And the biggest, you know, I'd say one of the trends I think we've seen in the U.S. in recent years around breeders and bull sales and things like that is there's more bigger breeders. You know, breeders that have a bigger sale with more bulls, and they've got kind of a network of cooperators that are feeding those in. And for the most part, they're pretty much all genotyped. I think as the price comes down and the accuracy where it is today, you know, it'll get to the point that's just standard business. I think it's good that pretty much every animal will be genotyped going in. That's my hope, or my prediction anyways.